To begin this procedure, the block of brain should be prepared for the cryostat and the section sampling frequency should be determined. Next, the antigen preserve and slides should be prepared. The brain block should be placed on the chuck inside the cryostat. Once the brain is securely embedded on the chuck, section at a preset thickness. Systematic sections should be taken such that series 1 is placed on a slide, and further series are placed in wells pre-filled with antigen preserve. Once the block has been exhausted, place the plate into a -20°C freezer as part of your brain bank.
Hi, I'm Dr. Mark Burke from the Visual Neurosciences Laboratory in the School of Optometry at the University of Montreal.
Hi, I'm Dr. Shahin Zangenehpour, also from the Visual Neurosciences Laboratory in the School of Optometry at the University of Montreal.
Hello, I'm Dr. Maurice Ptito, Director of the Visual Neuroscience Laboratory at the School of Optometry, University of Montreal.
Today, we will show you a procedure for systematically banking brain tissue in antigen preserve.
We use this procedure in our laboratory to maximize research material and as a basis for performing unbiased stereology.
So let's get started.
This protocol describes how to systematically sample brain tissue that has been fixed, stereotaxically blocked into 1 centimeter thick slices, and frozen. To see these earlier steps in detail, consult our recent JoVE article.